Alright. Okay, so, next lecture. Today, in this lecture, we're going to be talking about differentiation of cells and how we drive cells up and down the differentiation ladder, if you will, the landscape. The metaphor that's always used is like marbles rolling down a hill. And the hill, has different possible destinations, right? And to push it all the way back up from there to there is challenging. In addition, we're also going to talk about the early techniques and the later techniques for determining this is called de-differentiation. So for measuring de-differentiation ability of different kinds of cells in different animals, a lot of the work was done in frogs. So we're going to look at de-differentiation, title of the talk. Right, we're going to look at what it is, early methods, de-differentiation and de-differentiation. Later methods and human cells, but just the biological factors. By which I mean, what is it that keeps a cell in the state of being a particular cell, right? Because as I said before, all the cells have the same genome, and the situation in which a cell winds up is very stable. So if a cell winds up here, it's very stable in that position. But what makes it stable? What are the molecular mechanisms that make it stable in that situation? All right, so, like I just said, going from this situation, say it's a differentiated muscle cell, myofiber. I'm a big fan of myofibers. We need to take it back up the differentiation chain, differentiation pathway, in order to make a stem cell. Now, why would we want to do that? Well, getting stem cells out of an adult can be quite challenging, okay? You don't obviously have embryonic stem cells anymore. Getting stem cells out of your brain is fairly difficult, but what you have heaps of is skin tissue, is muscle tissue, and taking that tissue and de-differentiating it into an induced pluripotent stem cell, iPSC, which I think I've talked about before, is desirable because it allows us to take that tissue, de-differentiate it, and re-differentiate it into something new, right? So that's kind of what de-differentiation is. I'll talk very briefly on the mechanisms that control it. There's a thing called chromatin, which we'll talk about at the end. But that's a way that DNA gets packed up. When we pack up DNA, because there's so much DNA. DNA is massive, kilometers and kilometers of it are in your body. And in each cell, you need to pack it up really, really tight. And chromatin is one of the methods with which we pack up our DNA. And when you pack up the DNA, you pack up only specific parts. You don't pack up the parts that you use, but the parts you're not using are packed up and kind of inaccessible. And we're going to talk about that. Okay, so, part 2. Early methods for de-differentiation, and indeed determining whether or not you can use a more adult cell to produce an entire organism. So, what were the early experiments? Ow, I just threw that on my foot. Okay, so, early experiments. Right, so the early experiments were done with frogs. And what they did was, they took a cell at various time points in development. Right? So, at the time when it was a full-on embryo, like blastocyst, very early, right? It was quite likely, and then it was quite likely that you'd be able to produce a whole organism from a cell from that level of development. All right, so this is development. And this is probability of producing whole organism. Right? And so they took a cell from the developing organism at various points in development, and then they put that in the environment in which it should develop into a whole organism. So in much the same way we discussed the regrowth of limbs with salamanders, we just took a cell and put it in the environment where it should grow and saw whether it grew. And you very quickly get this drop off, right? It becomes totally impossible after like tadpole phase or something like that. And certainly not at swimming phase. So none at swimmers. Okay, and at swimming, not even possible at all. Right? So that's the early experiments, and we found that it was totally impossible to produce an organism after a certain level of development. That line looks kind of more like this. Right? So what were the later experiments? The later experiments were done using the nuclear material. So we take a developed cell, right? Like whatever, it's a cell. And it has a nucleus and it has its DNA in it, right? And we take that nucleus out of the fully developed cell, right? This is a fully developed cell. That's too else. It's a fully matured cell, right? And we take the nucleus out of it and we put that nucleus into the nucleus, into an egg. And we've removed the nucleus of the egg. Alright, so this is our egg, and into the egg we place this nucleus. Now we've already removed the nucleus from it. Can you see that? I'll do it in black so you can see the green a little better. Alright, there you go. Right, so that's an egg. And egg removed nucleus. Right? Now that's a big deal, right? Because this nucleus of the fully differentiated cell has all of the mechanisms in place that control a cell and force it to be an adult cell. Now remember before what I said about that landscape of differentiation, right? This guy. So we've got a cell that's down here, right? It's fully differentiated. And we need to push it all the way back up. And the way that we push it all the way back up is by putting it inside an egg. Now we're going to talk about, like I said, the molecular mechanisms that force that change, but this was that experiment. Now, how did they verify that this worked? So they took the egg of a green frog, and they took the nucleus material, the DNA of a white frog, and so here it's purple, and they found that the frogs that grow out of this cell are white. Now that's a good control, because if the white frog DNA were not able to fully grow and proliferate, you'd end up with a frog that was green or dead. More likely not a frog at all. You wouldn't end up with anything, because we've actually removed the material from this nucleus, right? So we've actually created a white frog using the egg from a green frog, which is pretty cool. Okay, so what is the deal with human cells? Now obviously we can't do this with human cells because that's cloning, and people get iffy about the ethics of that one, and I'm not going to get into it. But in order to do this in a human cell, you would first have to destroy the nucleus of a human embryo, which is iffy, and then you would have to put the nuclear material of a fully differentiated cell in there, which is cloning of humans, which is iffy. And we don't do that. That's a no-no, right? Keep that in mind if you ever get the idea of cloning like this with human cells, and then don't do it. All right. So, there are ways that we can test this. There are ways that we can test this with human cells. You take the nuclear material and you just see if you can differentiate it in a different environment. I won't go into those because it's not really part of this. But also, I'm going to look over there. I'm going to talk to you a little bit about the electrofusion approach. No, I'm not, because I don't understand it. I don't care about it. All right. So, as I said, I'm now going to talk about the molecular mechanisms. The molecular mechanisms, right? Right? So there are 4 molecular mechanisms that contribute to the state of a cell. 4 molecular mechanisms that make a cell in your arm different to a cell in your pancreas, right? As we recall, all cells have the same genome, same DNA. So the things that make cells different are mRNA, messenger RNA, microRNA, DNA methylation, and finally, histone regulation. Right, now remember I talked about those eggs where we've taken the egg from a green frog and put the white frog's DNA material into it. In that situation, the 2 bodies are kind of at war the whole time, and you'll actually only get like 1 cell differentiating properly out of 100. It's pretty iffy, right? These are the mechanisms that go on in the nucleus that we've put into that cell, that need to be reprogrammed by the egg. Right, I'm going to draw it again. Okay, so we've got our egg. I'm going to turn off Facebook, and it's got its DNA, right? Okay, one sec. Okay, so we've taken this nucleus out from a random cell and we've put it in this alien egg. We put it in this alien egg, not actually off the earth alien, just a different kind of egg. And this egg wants to turn into a whole organism. It wants to be pluripotent. It wants this nuclear material to divide and grow and turn into all different kinds of cells, right? It wants to be pluripotent, and it wants to grow into a whole organism. And this nucleus, by contrast, wants to be a fibroblast. It's like, I'm a fibroblast, I've got the right mRNA and the right microRNA and the right DNA methylation, and I've got the right histone regulation. I'm a fibroblast. Go away. And this is why not many of these work. Because you've got a nucleus that says it's one thing, and you've got an environment for the nucleus that says it's another thing. And so we need to look at how that's overcome in order to have successful cloning in this case, but reprogramming of the DNA. And let's look at the molecular mechanisms that are. So everyone knows what mRNA is, messenger RNA. We'll have a look at microRNA later. DNA methylation. DNA methylation was what I alluded to earlier. So, you have your DNA. And you have your chromatin. And chromatin is the little ball things that the DNA wraps around to compact the DNA. If you have an open chromatin structure, the DNA is less efficient to be read in the transcription process. It's bigger, it's more ungainly, but you can actually read all the genes. You can access all the genes. By contrast, if you have a closed chromatin structure, it's smaller and more efficient, and it's better to read the specific genes that you want, but of course you can't read all the genes. So, let's draw an open chromatin structure. Open chromatin structure. Right. It's going to look something like that. Open chromatin structure. Then if we have a closed chromatin structure, like some of the chromatins are still going to be nice and spread out, but others are going to be really on top of each other, right? So, say for example, this is the gene for something that allows us to be a pluripotent cell, right? Something that MyoD attaches to. We need a transcription factor, and this is the next point. We need a transcription factor to come in and activate those genes somehow. Now, a transcription factor that can do that is called a pioneering transcription factor. And I think I've gotten a little bit off track here. Now, let's forget about the egg for a little bit and talk about just reprogramming any cell from your body to being a pluripotent stem cell. We still apply the growth factor stuff. It's just that these transcription factors are likely to be present in the egg. But if we want to induce pluripotency of any kind of cell, fibroblast or whatever, you need to bring about the right transcription factors in order to induce pluripotency. The right transcription factors to induce pluripotency. And this is important. So the transcription factors that we need to induce, and it's amazing, it's fascinating that you can take just 4 factors and they'll influence maybe 25,000 genes and take any cell in your body and turn it into a cell that can grow into any other cell in your body. Like, isn't that crazy? Right, so you need some OCT4. That's the transcription factor. MyoD. SOX2 and KLF4. Now those transcription factors go into your cell, they activate a bunch of genes, which activate other genes, which creates a signaling cascade, and some of them, MyoD especially, is capable of opening up closed chromatin structures, which is really important for the differentiation or de-differentiation of these cells. And that, my friends, is de-differentiation and the creation of induced pluripotent stem cells.